There's a variety of mouse models applied in the CAR T-cell field that each have their specific advantages and disadvantages. One very frequently used model is the cell line-derived xenograft, in which patient-derived cell lines are engrafted into immunocompromised mice. The advantage of this model is that the cell lines are established and there is an easy access to these cell lines. Also, the growth of the tumor can be very easily monitored in case they are subcutaneously injected, so they can just be calipered. Also, the cell lines can be engineered to express luciferase, so then via bioluminescent imaging, the growth of the tumors can be monitored. Another advantage is that via genetic knockout or knock-in, the target antigen can be expressed or not expressed. So these cell line-derived xenografts are very useful, for example, for the study of novel CAR T-cell targets. Another frequently used model is the patient-derived xenografts, in which patient tumor samples are engrafted, again in immunocompromised mice. And then these tumors are passaged rather over mouse generations than in the cell culture. The advantage of this model is that the biological and the histopathological features of the tumor are better retained compared to the cell line-derived xenograft. The biggest drawbacks of both xenograft models is that they are engrafted into immunocompromised mice. So there is a lack of an endogenous immune system. And here, humanized mouse models come into play, in which an endogenous immune system is present, and the interplay between the tumor cells and the endogenous immune system and the applied cellular immunotherapy can be studied. However, to generate these humanized mouse models, hematopoietic progenitor cells are required. These cells can be either found in human fetal liver tissue or umbilical cord blood. Also, less pluripotent cells can be found in the peripheral blood of adults. Especially for adult patients, we don't have access to fetal liver tissue or umbilical cord blood, so we can create humanized mice using the peripheral blood. However, generating the humanized mice with these cells leads to usually to a bad reconstitution efficiency, and also graft versus host disease is rapidly induced.